Section 3. The LNPs, the known and the unknowns. Sorry for the slight delay of part 3. I had to put this series together much tighter as an entire 45-minute lecture in one go and record it. As you can probably hear, there's a good news. The quality has improved significantly. Thank you for that, my dear Nicole. So we can finally start. Yay! Will be fun. Promised. So up to this point, we've dealt with the membrane properties and the signal transduction that follows, and have already shown what a lousy idea it would be to tinker with this fine-tuning in a brute hammer meets with watch movement fashion. This brings me in the most important section where I will go into underlining my hypothesis with literature and questions about the LNP. Before we dive into this really brutal section, we need to clarify what pKa and zeta potential stand for. pKa is the acidity constant that becomes important during transfection in the cytosol, as the charge of the cationic lipids, according to the imagined principle, should be first neutralized in the cytosol. It is calculated in half of the charge loss. The zeta potential describes the total charge of the LNP on the surface. Quote, the layer typically referred to as the slipping plane serves as a boundary between strongly bound ions and bulk solution. The potential measured at this boundary is defined as the zeta potential. I will divide this section into 3 parts: signal transduction, direct and indirect evidence, which target cells, biodistribution. Signal transduction. In order to understand the current lipid nanoparticles, one has to follow the literature of liposomes, as the currently used formulations are in sum, to keep it simple, the much more complex further development based on this technology. It was and is exclusively about forcing an increased yield of proteins, miRNA, DNA, whatever is packaged into these carriers, forcing the endosomal escape better and more efficiently, controlling the biodistribution better, and increasing the transfection rate. Anyone who would claim that it was not known for a long time that the problems about signal transduction I discussed would be a bitter reality is either knowingly lying or has not followed the literature closely enough. Lunes et al. Published 2 very exciting reviews in 2008 and 2012. I will quote from the paper of 2008 2 short passages. Quote, however, increasing reports suggest that cationic liposomes strongly interact with the cell surface and modify the composition and the charge of the membrane. Later in this expansive review they write, a non-exclusive alternative to the receptor hypothesis could consist in a modification of the membrane environment of crucial membrane proteins upon insertion of cationic lipids in the membrane B layer. In that case, effects of cationic lipids on cell signaling or cellular functions would be directly related to their ability to fuse with the cell membranes. Fusion, lipid mixing with the cell membrane would be a way to insert cationic lipids in the lipid B layer of the plasma membrane and or intracellular membranes, and to modify the lipid-protein interactions involved in the functioning of membrane proteins. The ability of cationic liposomes to fuse with intracellular and plasma cell membranes, also with variable efficiencies, was demonstrated by confocal microscopy or FRET, first resonance energy transfer measurements, even if an endocytosis-like mechanism has been proposed as a main pathway of internalization of cationic complexes. In 2012, Lunes et al. Followed up with another paper that supports the theoretical framework we just examined in more detail. However, we now face a problem with lipid nanoparticles, as they have a significantly higher transfection rate, and we don't even fully know their chemical properties, including pKa and zeta potential, at last to my knowledge. Swanson and Erminova, however, calculated the temperature and energy dynamics and made an interesting remark. Do you remember when I asked you in section 1 to note the reference to the hydrophobic center of the B layer? Quote, both ALC0315 and SM102 can easily reside in the center of the lipid bilayer. End of quote. This raises a scary question when we consider that these LNPs consist of 4 different components: constant cationically charged lipids, phosphorylated lipids, pegylated lipids, and cholesterol. It can be assumed, I have already discussed this problem intensively with Maria Gucci, a pharmacologist, that one or the other cationic lipid may be dissociated during the transfection. How long would it remain in the membrane? Would it even be accessible to phospholipases? And could phospholipases deal with cationic lipids? Hands up anyone who gets goosebumps at the thought of what this means for the membrane-bound receptors, enzymes, and ion channels. Sorry, autistic humor. And here's just another paper showing nuclear factor kappa beta activation by empty lipids. What is remarkable about this paper is that it contains only this abstract and there seems no way to get the full text. And it was funded by the Defense Health Program. Nuclear factor kappa beta is one of the most complex transcription factors and its interactions are very, well, let's say, specific. Sadly, only a handful paper ever dealt with the consequences and looked directly into the problems of signal transduction and activation of transcription factors. So, I would classify the observations available on molecular biological level as indirect evidence of which I am well aware and can clearly identify that the MAPK, JAK-STAT, mTOR, CAMP and other signaling cascades are previously involved. A little fun fact about CAMP because it's super fascinating. By 2022, we found more and more evidence that AMPK cannot be considered independent of CAMP signaling and that it is another incredibly complex and interlocking cascade. In this regard, I would like to draw your attention to the paper by Deubner et al. It is one of the most comprehensive in vivo studies conducted on C57BL mice, so B6 mice. The mouse model is a good choice, as it expresses toll-like receptors and represents a somewhat predictable model for humans regarding inflammatory processes. The limitation is a 1000-fold too high dosage, but this is a typical way a toxicological study goes to observe the maximum damage potential, so it is a classical toxicological study. Keep this in mind. This is one of the most expensive experiments where we can see already the damage potential of the LNPs. I have marked some truly alarming observations that emerged in the GZA analysis. The TCA cycle, so the Krebs cycle, interleukin 17, one of the most highly inflammatory interleukins you can express. The PPAR gamma, peroxisome proliferator activated receptor gamma. They observed more than 9,000 gene expression alterations. These included high levels of inflammatory, apoptotic and necroptotic signals, but were not limited to these. In this study, the authors, including our Nobel Prize winner Drew Weissman, showed that the empty lipids matured and activated dendritic cells, which, as a logical consequence, first requires a firework display of signaling cascades that run downstream incorrectly. They et al. Even found that in their in vitro experiment both bone marrow-driven dendritic cells and macrophages with cationic lipids underwent expression alterations, which means changes in the ratio of expressed genes, and even the entire MHC class 1 and 2, so the major histocompatibility complex, was affected. So what they et al. Showed here was that cationic lipids can transfect dendritic cells and macrophages and result in direct alterations in MHC class 2 in dendritic cells. Also, no direct alterations in expression were seen in MHC1. This will necessarily influence the reactivity of all dendritic cells and the maturation of immune cells. Dendritic cells are highly specialized antigen-presenting cells that mature the natural killer cells, the CD8+ cytotoxic T cells, and the CD4+ T helper cells, as well as the B cells. And an altered MHC-1-2 ratio already has an effect on overall immune maturity. It would also need to be clarified whether direct crosstalks between MHC-1 and 2 also take place. It almost seems as if the entire MHC-1-2 dogma is slowly disintegrating as well. These processes are not well understood, especially the fine-tune and bridges which differentiate MHC-1 and 2 in dendritic cells, for example. I cannot and do not want to go into the MHC problem in any more detail, as this falls into the deepest area of the ninth immunological hell and has to do with VDJ recombination. Analogy for antigen-presenting cells. Cell goes through the body and shows a mugshot. Do you know him? And the best joke is that we don't even know how exactly this mugshot is created and what leads to incorrect presentation. This makes you wonder what kind of madness would be happening in the CD8 plus T cells and the CD4 plus T cells and the B cells as well at that moment. Another very concerning quote from this paper is, unlike for BMDCs, that did not show any alterations in glycolysis, BMDM's glycolysis and glycolytic were increased in both unstimulated and stimulated conditions when exposed to 100 picogram per milliliter of cationic nanolipid carriers. However, exposure to neutral nanolipid carriers did not induce any alteration in glycolysis or glycolytic capacity in BMDMs regardless of stimulating. This gives us indirect indications of possible mitochondrial damage and impending Warburg effect. This means that tumorous conditions have maybe already been created. Unfortunately, time is advancing and I still need to discuss several aspects, so I will now briefly list a few more papers that should definitely be considered and further strengthen my hypothesis and theory. So the next question is, which target cells? I will go through this a bit briefly. The next problem, which is frighteningly apparent, is the problem of the host cell, trademarked by Pfizer. It is very difficult to track the transfection efficiency and most likely target cells, as it depends on the pKa value and the zeta potential. So I have picked out some examples of studies where it has been clearly shown that both endothelial and epithelial cells can be transfected. As already mentioned by Day et al., which I quoted a bit earlier, dendritic cells can also be transfected. Furthermore, since the LNPs also preferentially accumulate in the liver, can we assume that Kupffer cells will also fall victim to these unnatural fat bombs? Remember at the beginning of this section why I explained the zeta potential and pKa? Quote Yu et al. Moreover, oxidative modifications on the cholesterol tail enabled lipid nanoparticles to accumulate more in liver endothelial cells and Kupffer cells than in hepatocytes. So, the simple answer to the question about the target cells is, we don't know. To quote Maria Gucci, who helped me with the pharmacokinetic and pharmacological aspects in detail, LNPs are weird. Change one little thing, even a small change in molar ratio, and bam, you've got something else. Which gets us into the third issue under unknowns, biodistribution. Or how I prefer to say, size and load matters. Let me quote Hermosilla et al. With a paper analyzing the in-use stability of mRNA LNP COVID-19 vaccines Comirnaty, Pfizer and Spikevax Moderna, a comparative study of the particulate. The expired Comirnaty samples were also analyzed after dilution and the results are shown in figure 1b. All vials analyzed belonged to the same batch, FG4686, and were 1 month vial 1, 2 months vial 2 and 4 months vial 3 expired by the time of the analysis. The particle size profiles indicated the presence of 3 clear resolved populations. The most abundant with an average DH of 60 to 65 nanometer, accounting for over 90% of the total volume in the samples analyzed. A second population with a DH of 600 to 700 nanometer also detected in all vials on proportion of 5 to 10%. And a third population with an average DH of 5000 nanometer only detected in vials 2 and 3, 1.2% and 2.8% by volume respectively. So we have 3 different sizes of LNP. Sounds like a great idea. It would never be a fact that the ability to cross blood barriers, like the blood-brain barrier and the blood-gut barrier, depends on the size of those stupid fat bombs, would it? Do you remember the TGA data? They labeled only with a luciferase assay the mRNA, and I'm not even sure if this was a mod RNA, cause they normally use this word when it comes into play. So one can only wonder, since these are the results from mouse and rat studies and only tracked mRNA expression, where we would find our unnatural fat bombs. And what about biodegradation? Quote from Jorgensen et al. Tertiary amines are a vital part of the ionizable cationic lipids used for mRNA vaccines. However, they are neither synthesized from endogenous or natural structures, nor are they degraded into typical biocompatible building blocks. The cleavage of fatty acids from currently used ionizable cationic lipids renders these auxiliary agents per definition biodegradable. Since synthetic ionizable cationic lipid building blocks remain after this cleavage, as discussed in more detail in section 3.2, this type of biodegradation does not improve their safety. So, the phospholipases will fail. Phospholipases are enzymes that deal normally with phospholipids, but they are not trained for cationic particles. Having said that, and having just read through the madness of LNPs and their predecessor technology, I'd like to move on to this paper. And this paper by D et al., which you are currently seeing as a slide, next to another paper, is one of the reasons for this endlessly long and very rapid discussion, because I would like to draw your attention to a bitter reality. We are not dealing here with the classic patterns of organ damage. The transfection can turn out so differently that the neighboring cell is spared, and you still have to expect long-term systematic damage. This is why conventional therapeutic approaches are more than problematic. That is why the focus must also be placed on signal transduction and membrane integrity. And to sound completely unscientific for a moment, it's always good to back up your argument with the last 2 papers. So both papers, the D et al. And the neighbor paper, are telling you that non-linear transfection of specific organ cells will happen, which means that the whole idea of typical markers of organ damage and considerations of organ damage has already become obsolete. We have left the known scientific perspective. Sounds like a great idea.